So one of the amino acids out of those 20 that I was telling you about earlier is called cysteine. And cysteine actually has the thiol functional group as its variable or R group within its structure. So if you remember back to the structure of the amino acid that I showed you earlier, we talked about how it had the backbone, which was the two functional groups, the amine and the carboxylic acid, as well as a hydrogen attached to a central carbon, and then we had an R group. And depending on what the R group was, that told you whether it was going to be a particular amino acid. So was it going to be an alanine, a serine, or in this case a cysteine? So when we've got the backbone of the amino acid, this time it's going to be attached to a thiol group, SH, that corresponds to the cysteine amino acid, and that's the one we're going to be looking at in the next couple of slides. So we're going to consider when we've got two cysteine amino acids and they actually become oxidized in a redox reaction. So remember, oxidation corresponds to losing hydrogen atoms. So there's going to be another component that it's going to be reacting with that's going to gain those hydrogen atoms. And once the sulfur atoms have actually lost the hydrogen atoms within those cysteine amino acids, the sulfurs are then going to satisfy their bonding arrangements by creating a covalent attachment together, forming a new bond. And the bond it creates is actually called a disulfide bond. So let's have a look at how this process works in the bottom reaction here. So firstly we have two cysteines, so two individual cysteine amino acids, and it's reacting with a compound that called glutathione. So there's 2 units of glutathione here. GSH is a way of abbreviating glutathione. Okay, so in this reaction between cysteine and this glutathione containing compound, one component is going to be oxidized and one is going to be reduced. So this is your classic redox reaction. So what's going to happen to our oxidized cysteines? Are they going to lose or gain hydrogen? Well, they're actually going to be losing hydrogen. So the 2 hydrogens attached to the thiol functional group here, as part of that, are actually going to be donated onto the 2 glutathione containing compound on the right hand side here. Basically what this means is the sulfurs are now going to have one less covalent bond. So what are they going to do to make up for not having the right number of covalent bonds? Well, they're going to form a new covalent bond. And the new covalent bond that it forms is actually between the 2 sulfurs. And this is where we get the name disulfide bond. Di meaning 2, so it's 2 bonds, or 2 sulfurs connected by a bond. So the disulfide means 2 sulfurs and they're connected by this bond here. So by oxidizing the thiol functional groups within cysteine, we are able to create the disulfide bond. So you can see in this 2 glutathione unit compound, there was actually already a disulfide bond. But by hydrogen coming in and being added to this compound, we've actually broken the disulfide bond, and instead the sulfurs have actually attached to those hydrogen atoms. So it's actually split the 2 unit glutathione compound into 2. So we've basically got the reverse of what we had at the beginning. At the beginning the cysteines were separate, and then there was a glutathione compound connected by a disulfide bond. Now the product side, we've actually got the cysteines connected by the disulfide bond, and then 2 individual glutathione compounds. So let's have a look at how this works in an animation. So we've got individual cysteine amino acids and then a 2 unit glutathione compound. So first thing that is going to happen is we're going to lose the attachments between hydrogen and the sulfur within the thiol functional groups on each amino acid. And we're also going to lose the disulfide bond within the 2 unit glutathione compound. So there'll be a level of energy input needed to start this chemical reaction, but to allow these bonds to break. So we've got the bond breakage, and then after the bonds are broken, we then have the hydrogen atoms associating with the sulfur atoms within glutathione, creating new bonds, and now we've actually created thiol groups where I'm putting the boxes here. So the glutathiones actually now have thiol groups. So without the hydrogen, the sulfurs within cysteine are actually able to come close enough together to form their own covalent bond. And that now means we've got a disulfide bond between the cysteine residues. And that is the 2-amino acid compound. So this is a good way to link together two amino acids by having thiol functional groups actually forming this disulfide bond through this type of redox reaction. So I'll show you in a later slide that the formation of this disulfide bond can actually be really valuable to creating a structure of a protein, because it allows two amino acids that aren't necessarily next to each other in sequence, non-adjacent amino acids, to actually interact and allow the protein to take on a particular shape. So here we have the reverse of what I just showed you in the previous couple of slides. This time we're going to break the disulfide bond between two cysteine amino acids. So initially we've got a cysteine compound that contains two cysteines and it's connected by a disulfide bond. And they're going to be reacting with two glutathione units that each have a thiol functional group. So it's these hydrogens associated with that thiol functional group that are going to be lost by these glutathione units and donated to the cysteine compound instead. So again, this will happen via a redox reaction. So because the cysteine compound is gaining hydrogen, is it being oxidized or reduced? Well, it's actually being reduced. So that means that these glutathione units are actually being oxidized. So because they're losing their hydrogens, they're being oxidized. So you can see the consequence of these changes on the product side. By gaining the extra hydrogens, cysteine is able to reform the thiol functional group with the sulfur attached to hydrogen. And now we've got two individual cysteines because the disulfide bond has broken. Whereas for the glutathiones, they actually had to connect together, forming a disulfide bond because they didn't have enough bonds without the attachments to their hydrogens. So you can see we've basically just switched the position of the disulfide bond. It was initially between the cysteines, but by undergoing the redox reaction, the cysteines are split apart and now the glutathiones are attached using that disulfide bond. So starting off with the disulfide bond between the cysteines and the individual glutathiones, we then get bond breakage between the sulfur and the hydrogen in a thiol group and also the disulfide bond, which then allows atom rearrangement where the hydrogens are donated to the amino acids through reduction, creating the thiol functional groups, and then we get the association between the two glutathione units forming a disulfide bond. So you can see from these animations we've had a look at, through the loss or gain of hydrogen atoms, it actually has an impact on where the disulfide bond is present, because the disulfide bond can only form when the sulfur doesn't have a hydrogen attached. So by accepting the hydrogen atoms, the compound can no longer form the disulfide bond. So because the cysteines accept the hydrogen atoms from the glutathiones in this case, they have to break that disulfide bond, and this allows a new disulfide bond to actually form between the two glutathione unit compound here. Alright, so now we're going to have a little bit more of a look at how we can use this disulfide bond to connect different parts of a protein structure together. So remember what I told you earlier, that the shape of a protein has a very large impact on how it can function. So that means we need to have ways of the amino acids actually connecting together other than just the amide links that connect each individual amino acid together that's next to each other in the sequence that we see down the bottom here. For example, how would this amino acid at the end, how might it come and interact with this amino acid? And one of the ways that different amino acids that aren't next to each other in sequence, call these non-adjacent amino acids, they might interact through a disulfide bond. if they're both cysteine residues. So if we have interactions or chemical bonds between non-adjacent amino acids within a protein, these are the ones that actually contribute to the shape of the protein and the way it's arranged in space. So they're absolutely vital to the way the protein functions, because the protein can't have its specific function if it's the wrong shape. Remember, we talked about the drink bottle example before. It has to have the right shape to be able to behave like a drink bottle. If it was flat, it couldn't do its job. Just like a protein, it needs the right shape to do its job. If it has a different shape, it can't do its job anymore. So that brings me to this point here. Without interactions between non-adjacent amino acids, the protein chain would be a long string of amino acids with no function. So essentially, without these type of interactions that give the protein shape, the protein is useless in terms of being functional, because it has no function. So if you just have a straight chain of amino acids like this connected by peptide bonds, it's not really going to be able to do anything, because the amino acids aren't arranged in a specific way that allows it to be able to function, for example, to carry oxygen. It can't do any of those fancy things because the structure isn't there to actually be able to provide that. But if we do have these interactions between non-adjacent amino acids, for example, this disulfide bond that I've shown you here. So for example, if the amino acid in this position was actually a cysteine, and so was this one, we can actually get a new bond that forms between those two cysteine residues, being the disulfide bond. So notice that the cysteine thiol groups don't have hydrogen attached, so it would have gone through a redox reaction losing hydrogen to create that disulfide bond. So how are these two different representations of a protein different? One without any of these non-adjacent amino acid interactions, and one with the non-adjacent amino acid interactions. What's the difference? Well, the one that without the interactions is just a straight chain. You can imagine it can basically just flop around wherever it wants. It's not going to be held in one position. And that basically means that it's very difficult for the protein to really do anything, have a function. Whereas on the right-hand side, what about its structure is different? Well, because of this type of interaction that we see here, the disulfide bond, basically means it's locked in a particular position. These amino acids won't have as much flexibility to move around because this interaction between the sulfurs within the cysteine residues is actually holding it in position. It's giving it a shape. So instead of being a long straight chain, it's now folded around. So these types of folding interactions is what gives a protein its overall shape. Instead of being a long straight chain of amino acids, most proteins form like a ball-like structure. And there's specific locations on that ball-like protein where different components can interact or bind. And that's what allows proteins to have their function. So here's a short animation showing how we can interchange between a straight chain structure and one that has some more structure. So some important parts to consider here is we've got the peptide bonds connecting the individual amino acids, which are shown in blue, and we've got some cysteine amino acids with the thiol functional group coming off it here. So this one would be a cysteine, and so would this one. So what do you think is going to happen when we push play here and it forms a disulfide bond? Well, essentially, it allows the protein chain to actually loop back on itself and get to the position where the two cysteine residues are close enough to each other to allow the thiol groups to interact. So the next step is for the hydrogens to be lost, donated to another component, and that allows the sulfurs then to form a bond, which is our disulfide bond. And now that the disulfide bond has formed, basically it is held in that conformation. So it's going to be a lot less flexible and it'll be held with this particular shape. Obviously protein chains are much longer than the 10 or so amino acids that I've shown here, but this just gives you an idea of how different amino acids can actually interact together, even if they're not next to each other in the sequence of the amino acids to actually add to the structure of the protein. So I'll replay that once more. So to allow the protein structure to fold and give itself a shape, we get the movement of these amino acids so that the cysteines are close enough together to form that bond which holds it in that specific arrangement. So we've got another Participation Plus question here, thinking about how the thiol group relates to the function of the cysteine amino acid and also the formation of this disulfide bond. So again, these questions are really useful for the comprehension of what we've done in this session, so I really do encourage you to have a go at these and share your answers with the peers you have in your online cohort. We've also done the content now for revision questions 9 and 10, so please pause and have a go at those questions on the LMS through the revision quiz. So to sum up what we've done in part 3 of this session, we were looking at some of the functional groups that were important to protein structure. So we saw that an amide was where we had a carbonyl group attached to a nitrogen, and that could have a few different arrangements. So if it was a primary amide, then it had one single R group, and that corresponded to an attachment to one other carbon atom, not part of the functional group. And if it was a secondary amide, then there were two R group attachments, and a tertiary amide has the 3 R group attachments. We also had a look at a few interesting compounds. For example, urea has the amide group and is produced in amino acid metabolism as a way of removing nitrogen from the body to avoid nitrogen toxicity. So the urea can be removed from the body through urination. So amino acids do connect together via amide links to form proteins. So remember, proteins have 100 or more amino acids connected together, and they're reliant on this amide link to connect those amino acids together. So we had a look at how these amide links are formed. So it's actually an interaction between an amine and a carboxylic acid, which lose some of their atoms to then allow the amide group to form. So in this instance, the amide is acting as a connector, so it's a 2R group functional group. So the carboxylic acid from one amino acid will be interacting with the amine of a second amino acid, and then you get those two fused together, linked by the amide. We also had a look at amide hydrolysis, which is basically breaking down the amide. So we saw that by using some of the atoms within water, you could actually break the amide link and create a carboxylic acid functional group for one amino acid and an amine for the second amino acid. So essentially, amide formation is the reverse of amide hydrolysis. So the thiol functional group is an SH connected to one R group. And we know that a compound contains a thiol functional group if it has thiol at the end of the name. And we saw that because it's a 1R group functional group, it's going to be located at the end of a compound. So one characteristic of thiols is they have strong disagreeable odors, for example, the smell of a skunk. And we also had a look at how we can either form or break down disulfide bonds. So a thiol functional group can actually interact with another thiol functional group to create one of these disulfide bonds. So for example, if you have two cysteine amino acids with the thiol functional group, if they each lose a hydrogen atom through an oxidation reaction, then the sulfurs can then attach together via a covalent bond, which is actually the disulfide bond. And we can also break that disulfide bond down by adding the hydrogens back into to recreate the thiol functional group. And the last thing we had to look at was the role of the disulfide bonds in protein structure. So essentially, if you had these disulfide bonds forming between non-adjacent amino acids, it was able to contribute to the shape of the protein. So instead of the protein just being a straight chain, it was actually able to loop back on itself and give the protein chain some structure. And remember that proteins are very reliant on their structure for how they can perform their function. So without these types of interactions like disulfide bonds to actually give the protein chain structure, it will actually not have any function at all. So that brings us to the end of our organic chemistry section of the course. So we've been able to investigate the properties of many different functional groups, and we'll actually continue to see these functional groups in the biochemistry part of the course. So in the next series of lectures, where we're looking at macromolecules like carbohydrates, lipids, DNA and RNA, as well as proteins, we'll actually see these functional groups come up again and again, and we'll investigate their properties a little bit further there as well. So I hope you enjoyed the organic chemistry section of the course, and look forward to sharing the next section of the course with you as well. Good luck with your revision, and I'll see you next time.